Everybody say hi. Although, not very many people are watching these, so it's okay. But they're around anyway. You got them. And that way, if you're gone. Did you watch our videos?
Yeah.
You did?
Yeah.
Oh, God. Good. I guess you can watch them on like 2x speed. That might work. Makes it, my stories go a little bit faster. Yeah. I mean, one thing is when I was lecturing just to myself, I fly. I mean, damn, I cover a lot of material really quickly. I should start doing this more often. But no, I like standing around in front of people. Anyway, so we've been talking about these basics of cell biology. We're now going to start talking more about the cell biology as it pertains to prokaryotes and bacteria specifically. And so there we go. So we got some terminology that is important. We have the envelope, which is everything from the plasma membrane, the phospholipid bilayer, out. And then we have the cytoplasm, which is everything from the plasma membrane in. And, oh, hi. You're back with us. You got released from prison. Do you have like one of those wrist ankle things? Or an ankle bracelet that they can track you? No, they don't. Were you watching videos online?
Yeah.
And that must have been fun. As I said, we're going to try and meet up for lab on Wednesday. Okay? And I'll hand out lab stuff, or I'll move or drop it. Anyway, so we got the cytoplasm. And in a eukaryote, we have the cytoplasm, of course, as well, but then the cytoplasm in eukaryotes is much more subdivided. We have the idea of the nucleus, we have the mitochondria or chloroplast if you're a plant, we've got the endomembrane system, and we just don't have nearly that kind of organization in a bacterium. And so pretty much we can say it's cytoplasm or it's not. And everything outside is the envelope. As we'll start seeing, we should get into ideas of metabolic pathways and things. Having compartments is important, and bacteria don't have much in the way of a compartment. They got the cytoplasm, and then they got the little space between the plasma membrane and other stuff, the periplasm as it's called. But in a eukaryote, you have some really good compartments in terms of like the mitochondria and the vesicle system that bacteria just don't have. But it makes the topography of these guys pretty simple. And then there is a flagella coming out. Often we'll have pili or fimbriae as they're called. That not all bacteria have those, but it helps to stick to things. Some of the pili can actually end up making a cytoplasmic connection that we can get DNA transferred back and forth. We're not really going to get too much into that. So, all right. The cell membrane, we've already gone through in terms of it being the phospholipid bilayer. And this is an important thing to think about in that, A, it is sort of self-organizing in that we've got the hydrophilic phosphate head and then the hydrophobic tails, the fatty acid tails. What this does is it creates a pretty good boundary between the outside and the inside. And one of the things that's important, particularly in cell biology, is the ability of different things can diffuse across the plasma membrane, and some things absolutely cannot diffuse across the plasma membrane. So if you are some kind of, a molecule that has any kind of charge to it, forget it. You can't go through the plasma membrane because the phosphate layer has got some charge to it, but anything with a charge is not going to be able to transit that fatty acid non-polar stretch here. And if we think about stuff that cells want to take up, a bunch of it has got a charge. That's everything from sugars that have a really strong negative charge, amino acids, proteins, salts. There's a lot of stuff that you need to take up that's got a charge to it, and it can't get through the lipid bilayer. And so we're going to have embedded in the lipid bilayer a whole range of transport proteins, things that can bind or tubes that will allow the charged molecules to pass through. So this becomes a way we can begin to select what comes in and what goes out. Just as a little note, there are things that can diffuse across the plasma membrane. And these are nonpolar molecules. Things like, chloroform is pretty good, steroids, different kinds of oils. They can diffuse across, so you don't need to have a receptor for this. It's one of the ways we can also exploit the cell membrane to blow stuff apart. You can add chloroform, and that does a pretty good job of disrupting the cell membranes. For that matter, adding detergent will do a pretty good job of disrupting the cell membranes. Remember, we talked a little bit about detergents have the ability to solubilize lipids, and so you can, even with the phosphate layer here, if you add a nice blob of soap, you can get the plasma membrane to dissociate. Now, the other thing with bacteria is they're onto us. They know this is a problem, and so they actually have a pretty thick peptidoglycan sugar coating that keeps their plasma membrane from being disrupted too easily. But it is something we can do. So things can get in either by just diffusing across, which is another point to make, is that water, CO2, O2, these are molecules that don't have enough of a charge to interfere, and we pretty much think of them as being able to diffuse across the plasma membrane. Water sometimes can have some weird tricky bits to it, but for the most part we just think about the fact that water and your primary gases of carbon dioxide and oxygen, they are able to diffuse back and forth, so we don't have to worry about transporting them. We then have things like facilitated transport, where we've got a protein, and just as a throw out a term here, hopefully you've either seen this term or it's not too complicated, the idea of diffusion. And like in A&P, in chemistry, you come across this, I hope. Diffusion requires a diffusion gradient. That is, it's a movement of molecules from an area of higher concentration to an area of lower concentration. And it's a thermodynamic event. So stuff can diffuse if you can pass across the plasma membrane. Even if you can't pass across the plasma membrane, if you've got some sort of transport protein, it can also carry out diffusion, but in that case it's called facilitated diffusion or facilitated transport. And then we can also use ATP to actually pump stuff in and out against its gradient, and that's what's known as active transport. What this all means then is we can take a cell, put it into a media, and the cytoplasm isn't going to be contiguous with that medium. It's actually going to have a different set of ion concentrations, and we can begin bringing in metabolites and exporting out waste products. So the plasma membrane is a critical thing for life. Pretty much, if you don't have a cell membrane, again, you don't get placed into the category of being alive. I say that, I think, is that right? I'm going to stand by that. Now, as we mentioned, bacteria, kind of like plants, they've got a cell wall. And this is important for a lot of reasons. You may or may not know, we do not have cell walls. And so we are a bit more delicate when it comes to water and salt concentrations and things. Now, with plants and bacteria, because they've got a really stiff cell wall, they actually can take up water and pressurize themselves and push out against that cell wall. We'll get into that later. The main thing is, when we begin to break down the cell walls of bacteria, we have two types that are based on something called a gram stain. And the gram stain, which we'll hopefully get to next week, is a stain that depends on the wall being present in a gram positive. It excludes a dye, and if it's not present in a gram negative, it actually allows the dye to come in. So we've got these two broad classes of bacteria that we can categorize right off the bat. Are you gram positive or gram negative? And they both have cell walls, but they're a little bit different in the way it's organized. So you've got, in a gram-positive, a whole set of molecules that make up what's called a peptidoglycan. And it is primarily sugar and a little bit of protein cross-linked inside of it. You then have a space known as the periplasmic space. And then right underneath that, you got the plasma membrane. So a plant cell also has the cellulose cell wall. And it's actually arranged kind of very similar to this. So the gram-positive wall, man, that is a tough barrier. And so I'll just say from a molecular biology perspective, blowing apart gram-positive bacteria is a way bigger pain in the butt than blowing apart gram-negative bacteria. That said, the cell walls of both of them are pretty darn tough. And, you know, I am a molecular biologist, and so I use bacteria all the time for gene cloning and for protein expression. And my number one bacterium that I use is the gram-negative bacterium E. coli. And E. coli, even though it doesn't have the peptidoglycan, it is an incredibly tough bacterium. In my experience, if you want the bacteria to lyse, if there's like a protein inside of it that's really important to you, you can pretty much guarantee that the bacteria is going to laugh at you and, show you its rear end and paddle itself and tease you, and it will not lyse. And then if you ever have a bacterium that you really need to keep intact, it'll fall apart, because bacteria are always just kind of mean in terms to work with. I got some tricks I can settle their hash. Boiling bacteria work pretty well to break them apart. There are some enzymes like lysozyme that will begin breaking down the lipopolysaccharide. That can start to break stuff apart. But bacteria in general are awfully tough little fellas. They can easily survive passage through your intestines. So, bacteria may be small, but we shouldn't jack with them too much unless you know what you're doing in terms of teasing them and things, because they'll get you. All right, so a gram-negative doesn't have this massive candy coating, at least exposed in the same way. We have peptidoglycan in the periplasmic space, but it's not as tightly cross-linked and dense. We have actually an outer membrane that's got a range of different kinds of protein pores in it, porin being one of them. And then there's a whole set of lipopolysaccharide, a combination of sugar and fat and lipid coming out that is the coating. And I will just tell you that lipopolysaccharide is a major source of bacterial problems. There are a whole range of bacteria that have really toxic lipopolysaccharide. LPS, as it's known, is a bad deal. So you can have dead bacteria, but the lipopolysaccharide can still get you and cause all kinds of inflammatory response. Toxic shock syndrome comes from this kind of stuff. So even if the bacteria isn't there, the coating on the outside can still give you a fit. Bacteria-induced diarrhea often comes from just the lipopolysaccharide. So it's a fairly nasty material, and it serves as one thing with all these guys. It's going to be a pretty good net negative charge, and so they have sort of a static electricity kind of interaction they're going on with. You can do a whole lot with these different kinds of cell walls. So peptidoglycan, we don't need to worry about the exact structure, but it is a combination of a sugar polymer, in this case a sugar called N-acetylglucosamine, and we actually have N-acetylglucosamine in our bodies as well. Your skin and tendons and stuff have got a lot of N-acetylglucosamine sugar polymers coming off of it. You then also have a range of proteins sort of cross-linking this. So you have, we've got proteins, we have glycoproteins, that is a protein that's got a fair amount of sugar on it. And then you have things that are mostly sugar with a little bit of protein that are called proteoglycan, or in this case, peptidoglycan. You got the carbohydrate backbone, and if we think about this, this is actually a very similar kind of thing to gelatin. In that, one of the experiments we used to do in the playing with food class is you can take gelatin and you can put a little enzyme that breaks down protein, a protease. And by clipping the proteins, you actually can de-gel Jello. You can do a similar kind of thing, proteolytically treating bacteria that begin breaking down their cell wall. Now, an antibiotic that you know of is penicillin, ampicillin, amoxicillin. These guys, all the illins, they block the synthesis of cell walls. And what that means is the bacteria can't produce, when they replicate, they can't produce a cell wall. And they're subject now to osmosis, which hopefully you've had that as a discussion point, but regardless, we'll get into it later. The main point is, if they don't have that shell around them, they pressurize up and then they pop. I would stick my finger in my mouth and make a popping noise. But so without the cell wall, the bacteria are really, really delicate little guys. But with the cell wall, man, they're tough. So penicillin has, though, a little spot called the lactam ring that is broken down by an enzyme called beta-lactamase, and that's what renders bacteria resistant to penicillin and amoxicillin and all the different psyllins. So we'll be getting into antibiotic resistance later, but it's actually a pretty cool thing, the way antibiotics work. So in the case of penicillin, ampicillin, they work because they're blocking the cell wall. All right. This is actually a nice thing because the cell wall composition is absolutely unique to bacteria. So it's a great kind of target for antibiotics because you can take them and it is really not going to do much to you. So we got our illins, cycloserine, vancomycin is a fairly aggressive one, bacitracin, a number of different antibiotics, and you can generate antibiotic resistance to all of them, but there you go. So again, this notion that we want to think about antibiotics in the fact that what you're trying to do is kill a living cell, but not kill the host. Anything you can do to get at it, what is specific to the pathogen, is a good deal. And bacteria, we got a pretty good angle on it. You start getting yeast infections and eukaryotic infections, and it's not nearly as easy to knock them back. All right, so a little bit more lipopolysaccharide. This is the material that is really making a lot of bacteria pretty nasty. So it is the outer surface of the gram-negative cell. That's what the G- means. And it's got this whole set of sugars coming off. And then we've got fatty acids. And then these are interacting with the cell membrane. So this is the lipoprotein that is part of the lipopolysaccharide. And they got all kinds of different names as lipid A. And in general, these are referred to as endotoxins. And so this is a lot of what makes bacteria pathogenic, is the fact that they can generate endotoxin. So, everything from toxic shock syndrome to diarrhea comes from the endotoxin. And one of the things that means is, even though you kill the bacteria, you still have endotoxin floating around, and it is still doing all kinds of things. So if a person has a bacterial infection that's getting a little bit too far along, even with antibiotics, there may not be a whole lot you can do. In fact, in some cases, when you provide antibiotics, you're dumping even more lipopolysaccharide into the system. And so you can have a really big immunological response, kind of an overexcitement of the response because you're dumping all this LPS into the system. So it's a fairly, at least for our immune system, it is something that we really can light up and fairly nasty. So we have then these terms for some of the outer coating. Something called the glycocalyx, which is essentially a add-on that not all bacteria have, and it is referred to as a capsule if it's really closely attached. It can be a slime layer if it's not very closely attached, and you perhaps have seen, with nasty food sitting in your refrigerator that starts to get really funky, that is bacteria that's producing a slime layer. On warm days out in the river, you can have bacteria coating rocks producing a really thick slime layer. And it's keeping the bacteria on the cell surface. These slime layers, because they are primarily a bunch of sugars, they have the ability to hold a lot of water. And so they serve as a kind of a hydration system. They also can block the immune system from grabbing hold of the cell and immunologically protecting it. So all these different kinds of things that the bacteria have to maintain its structure and for its defense. So on to various kinds of appendages. Not all bacteria have flagella, but those that do might have the ability to move around. And so we have some bacteria that we say are motile. They actually will spread about. Others, you put them down and they're stuck there. And what you have is a really complicated set of proteins we refer to as a motor complex, but of course it is just a series of proteins binding to each other. And they hydrolyze ATP, and that causes change in shape of the proteins, which in turn causes this thing to start moving around. It's a whip. And this allows for kind of a spiraling screw motion. They can begin to move around. Other things that bacteria might have are these pili, or pili, also known as fimbriae. And you can see here, this is a light micrograph where we've got the pili are extending out, and they're allowing the bacteria to actually adhere to things. We can have a pilus that goes between two bacteria that actually has a slightly different look to it. And it's actually a tube that connects the cytoplasm, and it allows for the transfer of DNA. This is known as conjugation. You can think of it as about as close as bacteria come to exchanging DNA molecules, i.e. having sex, but not really. Let's see, pili on the bacteria that causes gonorrhea anchors the bacterium to the epithelial cells. So a lot of these different kinds of structures have evolved to allow bacteria to move about, to keep them adhered to the surface, different kinds of things. All right. So now, as we get ourselves from the outer surface and all that stuff inside, we're back to some cell biology that we can use terms that relate a bit back and forth between prokaryotes and eukaryotes. So there are not that many actual elements that are structures in the bacterial cell. They're not eukaryotes. They don't have a whole lot of stuff. But what they do have are ribosomes that you can see, and pretty much only with an electron microscope. And the nucleoid. And then, depending on the bacteria, they can have spores, endospores. They can have what are known as inclusion bodies, just a generalized term, for some kind of little storage molecule. And you have things like starch, denatured proteins, all sorts of stuff can be held inside an inclusion body. There's always the chance when you're doing electron microscopy that you can generate stuff that is somewhat of an artifact. And so it's one of the things that when you're looking at electron micrographs, you have to be thoughtful of the fact that you have dehydrated this thing incredibly, you've embedded it in epoxy, and that the possibility for artifact is distinctly non-zero. There is also, depending on the bacteria, you can accumulate different kinds of long chain organic chemicals. And so bacteria, is not like looking at animals. There are wide ranges of bacterial growth habits and things. Very often, the bacteria will begin producing large amounts of protein and kind of crystallize it into an inclusion body. They can do a lot of different things inside the cytoplasm. So it's not very complicated, but there are some things in there. The cytoplasm is pretty much a gel. The concentration of protein in the cytoplasm is probably higher than what you can generate in a test tube, just because of all the protein interactions going on. If we think about what's happening in the cytoplasm, it'd be very much like trying to move through a semi-solid jello. I mean, it is a dense material. And there's a lot of surfaces there. There's a lot of reactions going on. So there you go. We'll be getting a little bit into some genetics next week, I think, or I think probably between the next exam, before the next exam, regardless. But ribosomes, if you recall, are the main structure that is involved in the production of polymerizing proteins. Ribosomes are what read the RNA. And they are a major kind of organelle, if you will, inside a bacterium. I mean, if you're going to say the bacteria have any kind of organelle at all, it'd be the ribosome. And the ribosome ends up actually having a lot of commonality with our ribosomes. So pretty much eukaryotic ribosomes in us and all eukaryotes are very similar, and prokaryotic ribosomes are all very similar. And they still have some pretty good distinct differences, primarily in the size. At least that's the first thing you would see, is that the prokaryotic ribosome is just smaller than the eukaryotic. When you see this, these terms like 70S and 80S, this is a terminology that's used kind of old-fashioned now. It's referred to as a Svedberg unit. It's how far a material will spin down in a centrifugation gradient. And it's the kind of technology that we don't have a centrifuge that can do it on campus, and B, hardly anybody uses this anymore. But there are devices that are known as ultracentrifuges that can spin at, like 100,000 times gravity. We've got little guys that can spin like 16,000, but these are great big centrifuges. And you put samples in there, and you have some kind of solution that produces a density. You can even use like density gradients, where you have different levels of sugar, like a sugar syrup at different concentrations. You put your stuff at the top, you put it in the centrifuge, and you see how far down it spins in this gradient. And there's like a normalized way of doing this, and this is what is referred to as the Svedberg or sedimentation units. And so it's a measurement of the overall size. So you have a pretty big guy, a pretty smaller guy. They don't quite add up, so you don't end up with an 80S, but close. So you end up with a 70S structure there. And then the eukaryotic guy is a little bit bigger, so 60 and 40, and then it adds up. There are some differences sufficient in the ribosomes that we can have some antibiotics that target these guys. So there are antibiotics like streptomycin, kanamycin, a whole bunch of them, some I've never heard, and neomycin is a fairly common one. These are interacting with the ribosome. The tetracycline class, these are also blocking either the formation of the ribosome complex or inhibiting the ribosome from doing its job. And because there is sufficient difference between prokaryotes and eukaryotes, it works fairly well. Okay. So the cytoplasm, it's easy to think of it as, or a cell as a a water balloon or like a jello-filled balloon, but there is structure inside the cytoplasm. In a eukaryote, there's a lot of structure inside the cytoplasm. In prokaryotes, there's still some, in that there are proteins that are able to shape the cell. If you think about the fact that we already talked about cell shape, you can have cells, bacteria that are actually somewhat comma-shaped, and that happens because the proteins inside the cytoplasm are kind of shaping it and twisting it about. Certainly, there are proteins that we just typically call the cytoskeleton, and they're involved in cell division when you have to pinch off two cells. If you got like a wad of dough, you can pinch it off, and that's happening internally. As you've got proteins that are sliding around each other, causing a pinching action to happen. They can, in eukaryotes, they definitely play a role in cell movement. In some prokaryotes, they can too, where you can push the cell along by changing the density of the cytoplasm. They also can interact with DNA during replication. There's a fair amount of sort of structure that has to be maintained and interactions that are going on there. So, lastly, we've got the genetic elements. That is, where we carry our DNA. And, again, in eukaryotes, this is going to be in the nucleus. And you got the chromosomes. In prokaryotes, we have the nucleoid, which is essentially the DNA sort of floating around. And so it's not quite the same as cytoplasm that doesn't have DNA in it. It's got a little bit of different kinds of texture and is sort of doing different things. The chromosome, this is the site where RNA is being synthesized, and in prokaryotes, it's also where protein synthesis is occurring. It also can occur in the cytoplasm. When we start talking a little bit about genetics, we can see the difference between the two. So the nucleoid is the chromosome, which is a circular piece, but it's really very big. Plasmids can also exist in bacteria, and these are circular DNA molecules that are way smaller than the nucleoid. So the chromosome might be 1 million bases, and the plasmids might be somewhere in the thousands of bases, so between megabase and kilobase size. Plasmids are also circular, but they're a lot smaller, and they have the ability to be moved around between different cells pretty easily, and they can encode for a whole range of different protein products, genes that will be translated into proteins. Very often, this is where antibiotic resistance will lay, and that a plasmid, rather than being actually in the genomic, the nucleoid. And so you get antibiotics, different kinds of enzymes and things, and things we think of as like sort of handy genes to have around that haven't yet incorporated themselves into the nucleus or the nucleoid. And plasmids can be moved around quite easily. The plasmid, because it's pretty small, can actually transfer between cells pretty darn easily, whereas the nucleoid is a lot bigger, and you have to have the sex pili and the idea of being able to have conjugation occur in order to have that DNA moving around. So plasmids are a critical part of thinking about how DNA is moving around. Let me finish this up here for the last bit. Another thing that bacteria are able to do that kind of belie their simplicity, we think of them, or I think of them as a dot, but the fact is they actually can work communally in that there is this notion of chemotaxis, in that the bacteria can actually respond to external stimuli. Specifically, there are chemicals that the bacteria will either move towards or move away from. So you can have positive chemotaxis or negative chemotaxis. And they've got a range of different movements. The flagella can sort of cause tumbling. And this is not going to be a particularly directed way of moving. They can pretty much just spin around. But then if there is some kind of chemotactic signal, they can begin to coordinate the flagellar movement and start actually moving in some kind of specific direction. And so if you tag a bacterium and you sit there and take time-lapse pictures of it, if there's nothing around it that is stimulating it, if there's no kind of chemotactic molecule, you just sort of have a random motion. But in the presence of chemotaxis, and this is a positive stimulus, you actually see that it's kind of playing the warmer, warmer, colder, colder sort of move, and it is zigzagging its way towards some target. So bacteria, they're single cells, but they have the ability to actually work in a somewhat communal, not necessarily like a tissue, but they definitely can work as a community. All right, we're going to knock off here and see where we go on Wednesday. We're going to have our first lab on Wednesday. I'll be giving lab manual thingy-mabob to you.